Hello everyone. This is the extra recitation 2 days before your exam. What we're going to do, so yesterday we went over exam, exam from 2013. We finished that. We we had just begun 2014, but we ran out of time. So today, just covering the rest of 2014 and then 2015. And if we have time by the end, you can just ask questions to your heart's content. So that should be fine. As far as I can remember, we reached the seventh question on. So this is the exam from 2014. Um, so the following figure shows the change in frequencies. As you can see, we have the allele frequency on the y-axis, p, and the time on the x-axis is just simply a time series. And p is increasing, as you can see, over time. And the question is, what additional information would provide the strongest evidence uh, that the change was driven primarily by natural selection, not genetic drift? So A, environment changes. B, environment hasn't changed. Population is small, population is large. Different trends are observed in similar populations experiencing different environments. So which one would be useful for us in your view? A, B, C, D, or E? So the environment. So environment changing or not changing. So the first two, they're not really useful. If the population is a small drift, is actually stronger. Again, as you can see, the probability of fixation is dependent on the population size. As the population becomes smaller, it's harder for selection to select for beneficial alleles. So that is a problem. So C is not the case as well. So between D and E, which do you think is the correct one? Population being large or uh, E, different trends are observed in similar populations expressed in different environments. So I think, in my view, so different trends are observed in similar populations. Muhammad says D. I believe that is the correct answer. So different trends in similar populations in different environments. That simply tells you that in different environments there are different either selective pressure or simply if it's drift, they are going to be different in different environments. Also going to be different in the same similar environments. So it's not really telling you a lot about what is actually happening. So that is not informative, but D is informative insofar as it tells us how things change. Okay, question 8. What a population shows an allele frequency at a frequency of p.4 and is in Hardy-Weinberg equilibrium, assuming that the entire population reproduces by selfing. What will be the frequency of the alleles in the following generation? Just to remind you, when selfing happens, so let's write it right here. So selfing again, the idea is or we use the example of two allele, two locus system that we've always been using. And in the case of selfing, A1, A2, so A1, A1, when A1, A1 mates with A1, A1, it only produces A1, A1, right? So that this allele is going to produce the same. A2, A2, when they self, they produce the same. So they produce the same genotype when they reproduce. A2, A2 also produces the same genotype. The difference, the problem is that A1, A1 would produce so half of the offspring going to be A1, A2, and the other half are going to be homozygotes, um, equally distributed between A1, A1 and A2, A2, right? So the frequency of A1, A1 in the next generation is going to be P squared plus PQ divided by 2. And A2, A2 is going to be Q squared PQ divided by. So that's the frequency of A1, A1 following the selfing, right? As we know, you can calculate allele frequency from genotype frequencies. So for us to calculate P in the next generation, so let's call it P prime, we should calculate if A1, A1 and if A1, A2 divided by 2, which is P squared plus PQ divided by 2 plus so the A1, A2 in the following generation is PQ again divided by 2. So P squared plus PQ divided by 2, which if I use a calculator, which I have, it should be 0.4, 0.4, that will be 0.16 plus PQ, which is 24. So the answer is 0.4. If you just calculate so using this equation, you get 0.4. That's the answer. So we simply took into account the property of selfing, which is heterozygotes are going to be reduced after each reproduction by half, and that half is going to be halved again, equally distributed between the two homozygous genotypes. All right, question 9. Familial FAP is a genetic disorder. Individual with FAP have a large number of polyps left untreated. Some of these initially benign polyps progress to malignant cancerous state when the patient is 35, 40. Consequently, untreated individuals with FAP have reduced lifespan. A major cause is occurring dominant mutations in APC tumor repressor genes. The mutation rate to FAP causing allele is whatever that number is. The frequency is this one, assuming FAP alleles are maintained in hematological mutation section. But what's the fitness of individuals? So it's a dominant allele, and the question is the selection balance. The mutation selection balance is roughly so for dominant allele that is not, so in this case it's not lethal, right? So it should be 2. We have Q and we have U, so 1 minus W2 is going to be U. How is it? 2 is going to be divided by Q. This one. So mu over Q is 4 zeros, yes, divided by right. So again, calculation. Okay, sorry, the frequency. Oh, sorry, 1 over 8. I made a mistake, so that's the other way around. 8 minus 1. And the answer is 0.87. Sorry, it should be 1 over 8. That's the mutation rate over the frequency. And I believe the answer is 87. All right. Let's move on to next. Okay. Imagine you take the sequence of 100 functional forces that we have been over these questions. So the idea being it should be second nature by now that you have the rate of non-synonymous sites. So the way non-synonymous sites change versus synonymous sites. So dn ds and depending on sorry, Alma asks about the previous question. Yes. So what was the problem with the previous? So the previous question. Here is the previous question, question 9. So if there was something unclear about this, please let me know. Did you normalize each of the individuals to each other? Like the fitness related to, well, the fitness, so the fitness of, so if we calculate W22, that simply is the relative fitness. So we really don't need to normalize anything. So as long as you just calculate this, so in this equation, if you calculate this, so in the case, so this is for the dominant case, right? Dominant non-lethal scenario. So as long as you calculate W22, that is relative fitness. So you don't really need to normalize anything. All right. And again, if you have any question while I explain or try to solve the question, please let me know. So we won't just move on without a clarification. So again, here we had the familiar question of non-synonymous to synonymous substitutions. As you know by now, non-synonymous substitutions, they have they can have phenotypic consequences, whereas synonymous substitutions, by definition, they don't really result in any change phenotypically. And thus, in this case, again, the gray bars represent pseudogenes. So these are genes that used to be functional, but they now they have mutated, so they no longer function. Versus the black ones that are functional regions, which produce proteins. So in the case of functional, or sorry, in the case of pseudogenes, it always should be the same rate as indicated in these two graphs, right? So because it shouldn't matter at all, you can accumulate substitutions that are synonymous and non-synonymous, neither of which are going to affect anything. In the case of coding regions, it's usually the case that you have you have negative selection, so you have more synonymous versus more non-synonymous. So D would be the case, unless unless it's the case that selection there has been positive selection. So if dn ds dn the S is less than 1, it's negative selection. Negative selection. If it's greater than 1, it's positive, and if it's 1, it's neutral. In the case of pseudogenes, it should be neutral, and that's why these the bar graphs here, in the case of pseudogenes, they should always be the same height. Okay, when the glaciers recede after an ice age and the species moves back into the once glaciated areas, the individuals that colonize the newly uncovered land or the leading edge of subpopulation near the limit of the species range during the ice age. Seeds contain mitochondria and disperse short distances. Pollen does not contain mitochondria and disperse long distances. Following the leading edge expansion, what patterns do you expect to find in genetic diversity in the newly colonized region when compared to the rest of the population? It's like increased genetic diversity in the newly colonized region. The differences should be similar in mitochondria and nuclear genes. Increased genetic diversity should be stronger in mitochondria than nuclear genes, or reduced diversity. So first of all, we have to establish if it's going to be increased. So we have to choose between A, B, or C and D. So which one should it be? Is it going to be increased genetic diversity or reduced genetic diversity? Anyone? Increase. So Sophians is increased. Almos is reduced. Christos is reduced. Shmita says reduced as well. So why reduced really? So it's like species moves back into once glaciated areas. So it's a founder effect, right? So you have a few individuals that expand in a or colonize a new region, right? So it should be reduced diversity, right, relative to the rest, because you have a few individuals who expand a new region. So it should be C or D, right? So between C or D, which one should we pick? So is there going to be the difference should be stronger in mitochondria than nuclear genes, or the other way around? Weaker in mitochondria. So we know that seed contain mitochondria disperse short distances, right? So they are going to show the strongest evidence because pollens are going to move longer distances and thus produce some level of migration, right? But mitochondria, they're not. So mitochondria should show a stronger evidence. Why weaker in mitochondria? So people who say weaker in mitochondria, can they explain to me why? What would be the reason for that? So the seeds contain mitochondria and they don't disperse, right? But the pollens they disperse, but they don't have mitochondria, right? So wouldn't that extend to reason that seeds should be less diverse, and hence and pollens are going to bring the nuclear genes from outside because of the dispersion. But mitochondria, they're not going to influence by that since pollens do not carry mitochondria. So the mitochondrial genes are going to show a stronger signal than nuclear genes, right? So there's an influx of nuclear genes, but not an influx of mitochondrial genes from the rest of the population. So it should be stronger in mitochondria C than. If anyone disagrees with this interpretation, please feel free to say so. All right, so let's go to what's the next question. Okay, so Holly says D. Why Holly? Why is this D? 10, 10 I actually I don't remember. 10 was B, yes. Okay, Holly also is fine then. So 11 C. Let's go to. Actually, let's check that just in case. Let's see if actually my interpretation was right. Finite populations. Okay, the founder effects. Okay, founder here, DNA, mitochondria. Okay, so yes, I was right. So this is actually, if you want to know more about this question, it's page 284. 284 in your textbook, where they talk about this example of the dispersal between in a plant when they discuss the founder effect. And so, using the same logic, they also conclude the same thing. So I was correct. The answer was correct. Two loci, A and B, located. So question 12. Alma says, sorry, let's go. Let's see this first. Yes, Alma's explanation is also correct. The word, yes, I mean, it can be slightly tricky, but again, be vigilant when reading these questions since the devil is in the details. Now, question 12. True loci. Layla says, what test is it from? It's from 2013. Two loci, A and B, located across chromosome 3 of the nematocilium, have two alleles, A and B. Following figure shows the frequencies. Which population would accept both A and B to increase in frequencies? So this one for heterozygosity, so 2014, so it's from 2014. So H prime is the haplotype frequency, so H prime of AB. AB is the haplotype frequency of AB. So these are actually so minus RD, right? So the linkage is equilibrium, and for the non-parental types, that's something like A, lowercase a capital B is H B in the previous generation plus RD, right? So using this, the only case, the only situation that would result in AB increasing, so we want H prime AB to be high or higher than HAB. We need and recombination rate cannot be negative, of course. So we need a negative D, so D should be negative. In order for HAB or H prime AB to be bigger, so D should be negative. And for you to calculate D, you just need to calculate the determinant of the matrix of haplotypes, which is actually just the frequency of parental types minus the frequency of non-parental types. Okay, so just calculate this for all these panels, and the one that has a negative one is going to be the answer. I think D should be the answer just by looking at. Well, actually, not AB. Yeah, I think D should be. Yeah, D should be the answer. D is the only one with a negative linkage disequilibrium. So just calculate the. You should calculate the linkage disequilibrium using this equation. Using this one, and because we want H prime AB, so haplotype frequency of AB to increase, we need a negative linkage disequilibrium. All right, let's move on to mutation accumulation. We don't know what that is. Okay, so heritability. Okay, resistance to West Nile virus in ravens is a trait that is known to have genetic component. We have been breeding ravens to have greater resistance to West Nile. It turns out you can estimate the number of West Nile virus particles in the bloodstream. When you expose every bird to the same amount of West Nile virus, some receive a larger infection. Well, allowing those that have the lowest infection levels to breed, you have developed a West Nile resistant strain. We're allowed to breed, average 40% lower infection rate than the test exposure. In each generation, you find that the average drop in West Nile infection level is 5%. What's the heritability? So what is the heritability? What do you think? So we have to somehow, so heritability in this case is narrow sense heritability. We want to see the response to selection is a function of heritability h squared multiplied by the selection. So the ones that we pick are 40% better than the average, and the drop is 0.5. So if we say 0.5, calculate, where is the calculate? 0.5. The average drops by 40%, also by 5%. So anyone with the question 14? Any suggestions? So the parents that we allow to breed, they are 0.4 better than the average of population, right? So the parents the response is a 5% decline, right? What is the irritability? I'm not sure what EE is. None of these are exactly. R is 0.05. Well, I mean, R is so every time. Because so, well, I would say E, but I mean it's kind of weird because so S squared 2 would be so 0.05 divided by 0.4 would be 125. I guess I mean this is pretty close to 1.3, but why isn't it exactly that? Hmm. Yes, I don't know. That seems like a very funky rounding. Yeah, that is weird. Uh, well, let me actually just so if there is so if this is not the actual answer, I'll post something on Blackboard. Uh, I'll check if that is actually the correct answer or it's not. Okay, so 15. Noted the size of the offspring produced by the willow beetle is related to the size of the parent. Measure the number of factors of interest. Mean size of a mid-parent and offspring willow beetle is 0.25 and 0.23 respectively. The covariance of mid-parent and offspring size is 0.72. The variance in the size. What is the irritability? So we have several numbers we have to parse. So we have the mid-parent uh, size. So this is this one, and the offspring is 0.23. The covariance of mid-parent and offspring is 0.72, and the variance in the size of the parent and offspring willow beetle is 1.2 and zero point 0.8. What is heritability of body size? Okay, let's move to the beginning when we have the equations to see which ones. Actually, it's not at the beginning; it's probably at the end. Okay, so let's look at the equations to see which ones are the ones we need to answer to this question. Okay, so the covariance of parent offspring, mid-parent offspring is this one. Okay, so one equation is this one. So the covariance of parent offspring. So we have that, which is divided by variance and mid-parent to the squared. Variance and mid-parent is again half of the, as you can see here. So variance and mid-parent is half of just parental variance. So parental variance divided by 2 would be variance in mid-parent. So we have a lot of actually a lot of things. So for heritability, I think 2 of them should be enough. I don't know why we have so many numbers. So we have the covariance covariance is covariance offspring and mid parent. So offspring mid parent. We have that is 0.72. The variance in size of the parent, so V P is 1.2. So the variance in midpoint should be half of that, so it should be 1/2 of 1.2, which is 0.6. All right. So where's my calculator? Okay, so if we say 2. Two, 1.2. So that's whatever the size. Interesting. I got 1.2. 1.2 is not what we want really. So what else do we have? Let's write down everything we have. So we have the variance of offspring, which is 0.8. We have the size, so the mean size of the parents and offspring, 0.2, 0.3. What if you divide that by 2 to get heritability? Oh, you mean 1.2 0.6? That would be odd though, because of okay, so when we go here, so the covariance between so offspring and mid parent, so this one, the one with the red with the green circle around it. So that divided by the variance of mid parent is 1/2 of the additive variance, so 1/2 of the parents. But then it says that the variance in mid parent is half of the variance in. Oh, sorry. Okay. So the mid parent multiplied by 2 would be okay. So now it makes sense. Now. I made a mistake there. Let's go back. How do we go back? Okay. So one thing that I actually made a mistake there was that it says that the mid parent is 1/2 the m. Okay, so VP would be 2. Well, that should be the case. What is that? We need to do that in that B equation. So what is the B equation? That's a little bit cryptic. It's B covariance. Oh, B covariance of x and y, variance of x. So half of the. Okay. So 1/2. So 0.6. So apparently, so if we use this, so the covariance of x and y, variance of x half of the heritability. So the variance of x and y in our case would be 72. The variance of x isn't that different? Not apparent. That is as what Jahat is pointing at. The that, that is the covariance between x and y. That's kind of funky. Okay, let me also I'll post this one on Blackboard as well because I need the exact correct answer for this. So what question was that? 15. Okay, let's just put that on Blackboard. So 15, 2014. So I'll put that on Blackboard as soon as I'm done with the recitation, so we can have the correct answer. Okay, so 16, you're studying the selection population of cheetahs. You measure running speed 4 months later. The average running speed has not changed, but that the variance has declined. Which equation would you use? So when variance declines, so if we look at the fitness function, fitness is W. So a divergent fitness function is the one where you're selecting for the extremes, and that one would not change the mean, but would increase the the variance should incline. Okay, so mean doesn't change, but variance declines. So we need something that is selecting for. So if it's the trait, so the histogram of the trait, we need something that is just selecting for the middle part, and so decreases the variance. All right, so it should be hyperbole. Sorry, not hyperbole. A parabola facing down. So facing down would be negative or positive. So it should have a negative second order, right? Coefficient, which is this one, right? Z squared should be negative. So it would look like a negative, or if a parabola that faces down. So D is the only one that is consistent with an equation which would characterize a fitness function such that you would only select for the intermediate values. All right, so 17, you're studying wing length in dragonflies. The range of wing length in your study population is 9 to 15 centimeters. You have said that dragonflies with longer wings are more successful. The primary food. Next generation, you again measure wing length and find that the range is now 12 to 18. The phenomenon is known as what is that? The latent variation. So it's not disruptive selection, of course, because they're not selecting for two extremes. It's not a stabilizing because it's just moving up. It's not fitness epistasis because you don't know what that is. It's not mission selection balance, so it should be latent variation. A few plants can grow in salt pans, areas of coastal marshes that have very high concentration of lithium, magnesium, potassium, and also skip 1920. Okay. Sorry, I did not. Okay, so if we skip these, then we can just move to 2015. Okay, let's find 2015. I'm sorry, what was 18? I totally Oh, sorry. I forgot the question 18 from 2014, which is our correlated response. Uh, they're two. They're genetically correlated. So selection in one of them would result in change in the other one. So these are genetic. And what was the 13? 13 we have to skip. You don't. Mutation accumulation is not part of the curriculum this year, so we don't need that. So 2015. So let's just quickly go over. So the ones that are simple, I'm not going to. I'm just going to tell you how to solve it, but we're not going to spend time on that. So in this case, so the first one, we just need to find a change that is synonymous. So just find the one that is synonymous, any of these four or so five. So the one that produces the same amino acid, that should be the correct answer. Two large subpopulations have the same spatial allele frequency of p 0.01, 0.7, respectively, assuming that both subpopulations are in Hardy-Weinberg equilibrium, have the same size. What is the heterozygosity in the global population formed by pooling the two subpopulations? So for each one, heterozygosity, so for population one, oh, sorry, yeah, one is for the terrorists, yeah, so they want the non-synonymous. So, I'm so sorry, non-synonymous mutation. So the one that doesn't code for arginine is the answer. So for population one, so pop one, heterozygotes are going to be 2 PQ, which would be 2 multiplied by 0.1 multiplied by 0.9, and for pop 2, it should be 2 multiplied by 0.7 multiplied by 0.3. Right. And then what you need to do is just calculate these two expectations. Oh, sorry, these two they're not expectation because they're in Hardy-Weinberg. So calculate these two, and then the average of these two frequencies would be the answer, which actually would be fewer heterozygotes that you would expect for the entire set of population. Which of the following statements true about rare alleles? So rare alleles is true. They tend to be beneficial, tend to be evenly distributed homozygous heterozygotes, so that can't be the case, right? Predominantly heterozygous individuals. So there's primarily, so rare alleles are primarily in heterozygotes. So it should be C. They can be beneficial or deleterious. They can't be predominantly in homozygotes because then why are they rare at all? For in class we was talking about natural selection. For example, we have explicit expression mean fitness. How does selection operate in this model? Every individual has the same fitness. Individual with certain genotypes live longer. Sexually, person with certain genotype produce more offspring. Depends on frequency of essential actually, in class, we're missing one particular model of natural selection in some details. For example, we drive explicit expression mean fitness. So this is A is not the case because then that wouldn't be a model of natural selection. It was not about living longer. Sexually mature adults produce more offspring than those with other genotypes. Okay, let's keep that. The depends on frequency. That wasn't the model. Zygote centrosomes are more likely to survive and reach sexual maturity than those of the. So what actually? So that's the questions that where selection was happening, right? Uh, which was zygotes actually surviving. So it was viability selection. So it's not C, it's D. So it was viability selection. The following figure shows question 5, hypothetical change in frequency, under which evolutionary scenario do we expect the frequency of the allele next generation to be 0.21. So in this case, so here at 0.2, it's more than, so we want a 1% increase. So here in B, it's more than 1%. In C, it's negative. In D, it's also negative as well. So the only one is A. Because at 0.2, there's 1% increase in allele frequency, which that's what we want. How many evolution scenarios shown in the previous question show a stable equilibrium in frequencies and allele between 0 and 1? So not 0 and 1, between 0 and 1. So there is, so B and C, they have a equilibrium at 1, so these two are already not relevant. Between these two, this one below, okay, so it should be 8, right? Because it's decrease. It would increase if it's below equilibrium. It would decrease if it's above it. So there's only one with a stable equilibrium. D is unstable. D is actually the underdominance case. Underdominance and thus unstable. Okay, this question 7, simple. It's just the changing population size. So just calculate the NE over, it would be T is the number of generations over 1 over N1 plus 1 over N2 plus 1 over Nt, which is, we should calculate it because you shouldn't just look at them and decide which one has the biggest effective population size. The calculation is probably the best way to go using this equation here. 8 following figure shows 4 hypothetical relationships between the fitness of genotypes X and its frequency, under which evolutionary scenario is the X genotype not experiencing frequency-dependent selection. So the only one without it is B, where it's just a line. So there's no relationship between the fitness and frequency in B. Question 9, industrial melanism. So the change, so they following industrialization, moth became darker. But since they've stopped doing that, the level of industrialization or pollution dropped drastically since the 60s. So what was the reason for it? Because melanic morphs declined. The melanic morphs, their fitness depended on the environment. As the environment changed, the fitness decreased. So it's B. It's not frequency dependent, really. And it's not Bolund effect as well. So TPI enzyme, so there's a lethal mutation, there's a dominant mutation. So the only thing you need to know is that the for question 10 is that the for recessive allele, the equilibrium for recessive allele, recessive lethal, the equilibrium is equal to the square root of mutation rate. but for a dominant, it is just the mutation rate, right? So for and so the square root is going to be a larger number because it's a number smaller than 1, so the square root of that is a bigger number. So the mutation rate down is equal to the mutation. Okay, so what was the question actually? So the frequency. So if the frequencies are the same, so the mutation rate to dominant allele should be higher. It should I think it should be. All right. I think one last question before we conclude. After that, you can ask questions. The following figure shows the measure of genetic variance in 17 islands. Galapagos says as the island becomes bigger, there's more heterozygosity. So A says the smaller islands contain large populations. That's not the case. Smaller island, large populations, no. So between the C, D, and E, so small population causes genetic drift to act less strongly, so that's incorrect. So it should be more strongly, so now we're between D and E. More drift is going to decrease genetic variation, so it should be D. So drift decreases genetic variation, not increases. For 10, can you explain why the machine is higher than done? Okay, so for 10, Sarah asked why is that. So let's say, so Q for dominant is, for the dominant allele is 0.1, right? So then the mutation rate for dominant allele is 0.1. If Q for the recessive is 0.1 as well, so it's 0.1 equals to the square root of, so it should be, actually, oh yeah, sorry, yeah, so let's calculate. So point 1. So okay, so the rate to dominant allele should be higher than. I mean, it should be higher, right? Because the square root of a what is the square root of actually? This number. What is the? Let's. Okay, square root. Okay, so the square root of a smaller number is going to give you 0.1, or so 0.01. So the mutation rate for recessive is smaller. So that's why it is B. Oh, okay. Sarah is also satisfied with this answer. Although I'll also explain the reason for it. So because it's a mutation rate is a number that is always smaller than 1, and that's why this happens here. All right then. So it's 4 o'clock. If you have any question before we conclude, please ask. If not, and this is going to be uploaded in a few times. If you want Luke, join our. I'm not really sure. I can't really understand that question. Okay, any comments before I just leave? Number 2 on the homework. Well, not now. Well, I'll probably just if I don't know if Diane is going to post the answers. So if not, I'll post an answer before the exam. All right then. Okay, have a I mean not a weekend. It's Tuesday, so good luck with the exam. It's going to be easy. There's nothing really there that you can't do. It's pretty basic population genetics. I think you can